So hi, hello and welcome again. Microbehunter here. Uh, yes, I think sound seems to be working. I'm just checking if everything's fine. The sound is good. Thank you very much. I did move my microphone a little bit further away today, and you see that the setup is also a little bit different. You can now see me on the other side, on the left side. Okay, because I rearranged the camera a little bit here on my desk. So, and I'd like to welcome you again to another, Saturday microscopy live stream. A week passes so quickly, it's almost unbelievable. I would like to say again hello around the world. The first couple of minutes, I usually wait a little bit until more and more people started to join in. The reason why I set up the camera a little bit differently today is because then when I'm looking into the screen, into my computer screen, I'm a little bit also looking a little bit more into the center of the video. That's basically the only reason. Otherwise, I'm looking too much off screen. Yes, I'm just quickly reading again what I'm able to see over here. Hello from Lebanon, from Belgium, from London, from the Netherlands, from England, from Württemberg, from Germany, Texas. Hello from Austria again. Hello from the Netherlands, from Colorado in the US. Okay, that's one of the things that I like. Microscopies enjoyed all around the world. Hello from Athens in Greece. I think now that approximately 1 minute or 2 minutes have passed, I think let me quickly check how many people are online. It's also sometimes interesting to see. Over 30 people, 35 people. It's quite nice. So what I'll be doing today is the following. As always, the first hour we're going to be doing some microscopy work. Then if you have any questions that are either on topic or off topic, please do feel free to post them in the chat. There is also the possibility to switch over from top chat to live chat. Okay, so this gives the whole thing a little bit in the real time. What I would like to also say is that if you have any off-topic questions, please also feel free, but I will answer them after approximately 1 hour. What will I be doing today? It's actually something that I've planned already doing for some time now. I've got over here a water sample which I collected a few days ago. And what I've done is I added those white things that you see floating in here, this one and this here. These are wheat grains. I just want to show it to you, just a second. So I've got some wheat grains also over here. And those wheat grains, they look like this. Yeah, let me refocus here. What I did is I crushed them between 2 spoons, and simply to expose the starch a little bit. I simply dropped a wheat grain then basically into the pond water sample over here. And this was, I think, about 3 days ago. And in the meantime, the wheat grain served as a food for microorganisms. And when I put a little bit of the sample today in the morning under the microscope, I could see that there were, of course, a whole bunch of ciliates present, but also some spiral-shaped bacteria, spirochetes. And that's actually quite interesting to observe. And then I decided, well, why not do the live stream today by testing out different disinfectants on whether they're able to kill those microorganisms. So I've got a whole bunch of disinfectants here, and that, like for example, regular hand disinfectant, based on hydrogen peroxide. I've got some alcohol here, some strong acid, and this one over here, this is garlic, because garlic itself, pressed garlic, also is able to kill off microorganisms, and I would like to observe this under the microscope. Okay, some safety precautions first. Okay, I just want to, before I go ahead, I just want to show you a little bit what I've got here. The liquid in here, that is alcohol. Right, and I'll be using those pointed tweezers to pick out the wheat grains which contain now the microorganisms, and then I will basically, after transfer them on a microscope slide, I will put it back into the alcohol. The reason being that, of course, there are now bacteria also on those tweezers, and those tweezers are quite pointed, and if there's an accident and if I puncture myself, then of course I'll be transferring some bacteria into my body. I do not want that, and especially those spiral-shaped bacteria that I found today in the morning. You do not want to get those into your body because some of them could actually be dangerous. Okay, and I don't know what they are, so for this reason you have to be extra careful. So this is a little bit the thing that I would like to do today. Okay, if you have any, and then later on, I also have another question to you, to the community, because I would like to also get your opinion a little bit on what videos I should make in the future. Okay, so what I'll be doing is the following. I will. For each disinfectant that I'll be using, I will be putting a small sample, a very small sample on a microscope slide. Cover glass goes on top, and we're just going to look at it under the microscope first. Okay, yeah, vinegar is also one of the things that I'll be testing. It's actually it's not regular vinegar, but actually it's quite strong vinegar. It contains, I don't know, I think over 20% acid vinegar, I think, basically regular vinegar has I think about 4 or 5%, but this one is significantly stronger. So let's do the following. Let's just get started. I do need some cover glasses as well. Where are they? I've got some over here. So here we go. Yeah, the reason why I got them in here, the cover glasses, is because I washed them, I reused them. This reason I've got them in here, but still they're not entirely clean, so I need to occasionally also dry wipe them. So this I will take out the tweezers from the alcohol, and what I have to do normally, and I'm not going to do that because of safety reasons, what you can actually do is you can use a lighter, and that's what we've used in the lab to burn off the alcohol. Right? I'm not going to do that because there are alcohol fumes here, and if I happen to spill it, then it's a danger of fire. So we'll not be using a fire, but rather I'm going to dry off the alcohol on this tissue paper. You have to dry it off because otherwise I'm going to transfer the alcohol into the specimen here, and I don't want that. So let's simply give it a try. I'm going to take this here. And I'm going to make a very small, and I transferred it back here. You can see that it's stuck here, not good. Well, actually thinking about it, because I'll be reusing it anyway over here, I might as well leave the tweezers in here. I think that's the easiest way. I think, I'm just saving, but didn't think of that. I'm just saving myself an additional step here. And I'm going to now put the, cover glass on top, and then we have to check if there's actually something of interest here. At least today in the morning, I was able to find quite a few things here. Okay, so greetings from the UK, I just read here as well, and also from Greece. So let's have a look. Okay, I'm going to put it now on the slide over here. Where is my scope, the microscope view? Let's turn up the light a little bit. That's the 4 times magnification. It's of course over here I have to, let's go up a little bit. I'm a little bit disappointed because today in the morning I really had a lot of microorganisms in here, a lot of bacteria, and I wonder, okay, well, here are a couple of ciliates. I'm a little bit surprised because there were so many spiral-shaped bacteria present, and I wonder maybe I picked up the other the other one. It doesn't matter, we still have a whole bunch of microorganisms here that we can test it on, but I'm still interested. So what I'm going to do is I'm going to simply try the other one. It's what you call demonstration effect, because I'm going to now check the other one and see if maybe they're slightly more present here. And another cover glass goes on top here. Okay, let's have a look. But in any case, you get the point that you can make an enrichment culture this way. It's very similar, but there seem to be slightly, I think that must have been the one. There's slightly more microbes here. Okay, let's go up a little bit with the magnification. And here we go. Yes, you see that there are a whole bunch of bacteria moving around as well in the background. So you see, it's kind of interesting that even though, it's in the same jar, the 2 wheat grains basically, it looks a little bit different. It almost looks like this guy over here is dividing. Okay, so you've got a couple of dividing cells here as well. The one here in the center now. So, and let's have a, wow, there's quite something going on here. Okay, quite nice. So I just want to quickly explain to you what we're able to see here. Maybe I just go up with the magnification yet a little bit further. Okay, yes, that's much better. Yeah, here are the spiral-shaped bacteria. So let me show you, where is the arrow? The arrow is here. Yeah. Okay, so you see those very fine lines here in the background. Sometimes there are those spiral-shaped corkscrew rotating. Yeah, these are bacteria in the background. Yeah, those large things, guys, these are ciliates moving around. Yeah, but these tiny, yeah, here sometimes spinning very quickly. These are bacteria. I do not want to get them into on my fingers, obviously, right? So, yeah. A little bit, some of them are a little bit out of focus. Those large things that you see here seem to be starch grains from the wheat grain. Yeah, so that's basically the food source. Yeah, also these things here. Yeah. Yeah. So yeah. So yes, there are some, it says here on topic, what is the alcohol percentage you're using to disinfect? I will tell you the following. I'm going to add some 97% alcohol. However, it's very important. The concentration of the alcohol does not stay the same because, of course, there is plenty of water here as well. And in the lab, when I still was studying at university, microbiology, we always used 70% alcohol. And the reason is because, interestingly, 70% alcohol is more effective than higher concentrated alcohol, and there's a reason for that. The reason is because the 70% alcohol is able to dissolve the cell membranes better. So that's the reason. Because if the concentration of alcohol is higher, then what's going to happen is that the cells will actually lose quite a bit of water as well. They might shrink. Because water is moving into the alcohol by osmosis, and then when the cell shrinks, it's not able to pop as easily anymore. Because what you want to do is when you treat the cell with alcohol, you want to dissolve the membrane so that the cell actually pops open. And actually, that's one of the things that I would like to show you. Okay, and 70% alcohol is actually the standard concentration that we used, for disinfecting surfaces and our hands and so on. Yeah, but what I'll be doing is I'll simply be adding concentrated alcohol, a small amount, because as the alcohol moves into the liquid, it will be diluted anyway. Yeah, so here we see again there's quite a bit going on here. Yeah, and demonstration. So what is this, sir? So for those of you that just joined in, this is basically a so-called an enrichment culture. I collected some pond water, added some food, some wheat grains, and waited for 3 or 4 days. And this is what we found. And what I'm going to be doing now is I'm going to be testing out different disinfectants. I'll be adding small amounts of different disinfectants, and I'm going to see what's going to happen. And I just need to also tell you a little bit of background here. When we're killing them, so to say, then of course they stop moving, right? So that's a good sign in this case. But just because something is not moving does not mean it's dead. So that's really important because, in this case it's easily visible, but just because something is not moving, it still could be alive. Maybe it's just a non-moving microorganism, right? So I'm going to also tell you a little bit. Yeah, here again you see those spiral-shaped bacteria that are moving like corkscrews. Some of them can be causing problems, right? So for example, Borrelia, and so on. From Lyme disease, these are also spiral-shaped bacteria. So generally, you got to be careful anytime when you are dealing with unknown microorganisms. Yeah. So what I would like to simply also make you a little bit of aware what I'm going to be doing so that it's easier for you to understand. So let me go down a little bit with the magnification and also the brightness. I'll be using my pipette to add a little bit of alcohol or the different disinfectants to this sample. And what's going to happen is the following, just that you're aware. The alcohol will move beneath the cover glass, and it's going to start flushing the specimen, and there's going to be a stream of liquid. Yeah, so this basically means that many of those cells, or almost all of the cells, will be kind of pushed to the side and are going to move out of the field of view. So what I have to do is I have to kind of change the position of the slide again to find them again, and then we're going to see if they're moving or not. Yeah, but don't be surprised if many of them actually are going to be flushed away because me simply adding the disinfectant. Yeah. So yeah, some of you are already identifying what they are. Okay, thank you very much. Yeah, so that is simply something that I would like to do. What I'm going to do now is just for your info. I'm going to turn down on the camera. It might look a little bit darker now, but the how do you call this, the frame rate is a little bit, the exposure is a little bit shorter, so the movement might be a little bit better to see. Also, when I'll be adding, look at the top, okay? I'll be adding the alcohol over here, right on the edge of the cover glass. Of course, I have to be extremely careful because I do not want to get any alcohol between the objective and the on the objective. Yeah, so I have to be kind of a little bit careful here, and that's kind of the idea. So I'm going to now do the following. I'm going to simply show you what I'm doing on my table before. Okay, this is a question about the forum. I'll respond to that later. Okay, so here's the desk view again. So let's focus again. I've got a nice camera here. Yeah. Over my desk. I'll be adding a little bit of this is just regular ethanol. Yeah? I think it's 97%, 96 or 97 percent. That's the standard one, fairly cheap to obtain. And rubbing alcohol, isopropyl alcohol should also work, and that's my pipette. I'm going to, I don't know, why not standardize this? I can change the volume over here. I don't know how many. Shall we just try 20 microliters? Let's see if this. Yeah, it's still quite a lot. Yeah, maybe let's just try 10 microliters. Yeah, and so here it is. Back to the microscope view. Here we go. So, and let's see what happens. Okay, so I'll be just also look on the small video that I have up here. Yeah, and at the same time also look a little bit at the cells on the side. And as the alcohol goes beneath the cover glass, you should actually see them being flushed away. And then we have to try to quickly find them again. So, okay, they're actually not so much flushed away, because but the other bacteria are. So we have to now wait a little bit until the alcohol reaches those cells. Here, it's a place already. Let's try to find a place where they're still moving. Okay. And I was actually, here they're still moving. And then I just want to show you a little effect that the alcohol has, because if we're lucky, we're actually able to see that the alcohol dissolve the cell membrane away. Look over here, they're already dead. Down here, they're still moving around. I do not want to go up higher with the magnification because I'm a little bit worried that my objective becomes, but there is actually no liquid on the microscope slide itself. Yeah, so, okay. Here they're still moving. Let's add a little bit more. But look! Do you actually notice? Look at those. Look at those. Where is this? Look at those spiral-shaped bacteria in the background. They stopped moving. Yeah, most of them. Yeah, those spiral-shaped bacteria. So actually, the alcohol already had an effect on them. Yeah. But maybe not yet on the other cells. And over here, look. Here the alcohol already had its effect. So let's do the following. Here we are. I'll add a little bit more. what you are able to sometimes observe is that if there is a gradient of, let's say, salt or a gradient of alcohol or gradient of anything which is unfavorable, then they will actually move away from this from the higher concentrated area to the lower concentrated area, and this also allows you to kind of concentrate them on a microscope slide. Yeah. Will I try soap? Oh, actually, I wanted to try soap, but I forgot to prepare the soap. Okay. So yeah, what we see over here is of course you see that there is this large clump of yeah, material over here which kind of shields the microorganisms from the yeah, from the alcohol. Let's add a little bit more. Okay, and yeah, they're of course they're trying to move away from the alcohol. Let's have a look here. Yep. This one over here is not feeling so good anymore. It's starting to go a little bit uncontrolled. And actually today in the morning when I tried it, I was quite lucky because I could actually see that the cells actually popped open and the cell's contents were spilled. It's actually also the thumbnail that I showed you in this video. Yeah. Okay. Yeah. So this one definitely, but let's zoom in here a little bit more. Let's make it a little bit brighter. So that's one of those. And if the concentration is sufficiently high, then we should actually also see the alcohol dissolve the cell membrane. I don't know if we're able to see it here. Yeah. But you can also see that the other bacteria over here on the left side, the one over here, yeah, is also already affected by the alcohol. Yeah. So let's remember this one here. I've also seen in the past that not all microorganisms are equally sensitive. Some of them, like to lice, pop open quite easily, others not so much. Ah, yeah. Look, something seems to be happening. It seems to be kind of, yeah, bulging out here, which is a sign that the cell membrane is already starting to dissolve. Yeah. I don't know if I can go up with the magnification a bit further still. Yes, so that's now 60 times. There are still some bacteria there. Okay. Alcohol, as a matter of fact, has a strong tendency to dehydrate the specimens, to basically also pull the water away. So the right concentration of the alcohol, of course, determines then a little bit of what's happening to the cell, whether it's actually going to pop open or not. So very quickly, have you ever encountered a gyrus? I suppose you meant to say virus. And as a matter of fact, viruses cannot be seen using light microscopes because they are too small. So that is, well, I can talk a little bit about that. It is theoretically possible if you increase the contrast by using so-called dark field, you are able to actually see viruses as small bright dots, but you're not able to identify them as such, because you just see a bright dot and then you don't know what it is. But look at this cell now, okay? I think, yeah, you can now see that it's completely lost its original shape because the alcohol has done its job and started to dissolve away the cell membrane. Yeah? And, yeah. I was actually talking about the giant viruses. They can be as big as 400 nanometers. Oh, okay, no, I've not seen it. Yeah? And in some cases, it could be that even though they're quite long, maybe sometimes they're very thin so that they cannot be seen. I don't know about it. Okay, but I would have to have a source of those. So let's basically have a look here again. So you see that the base, what the alcohol does to the cells. Let's see if there are still a few of them who've survived down here. Yep. Here on the other side, the alcohol did not reach the cells yet. Okay. So And yeah, look, these two cells here also seem to be a little bit connected. So they're in the process of dividing as well, apparently. It's also quite interesting. Or conjugating, I don't know. Conjugating means that when the cells are joining together to exchange DNA. This does not look like a division. If the cells divide, it looks more symmetrical. Maybe we are observing a conjugation. I don't know. It looks like it almost. Yeah. Interesting. Yeah. So when ciliates, when they conjugate, what they do is their membranes fuse together and they exchange DNA to increase genetic variability, because they, of course, they divide by cell division, obviously. So how do you introduce genetic variability now? And they do that by exchanging DNA and mixing it this way. Yeah? That's also quite nice here. Yeah. So some comments here. I've recorded several ciliate deaths on my live streams by accident. It's amazing how they can keep swimming as they break down. Yep, that's the often stop contractile vacuole CV pumping first. Yeah, so what I've also seen is sometimes that indeed the cilia, the little here, they continue to pump and move, not pump but move, even though the cell is already breaking apart on a different place. Yeah? So that kind of shows also a little bit that they're a little bit autonomous. Yeah, the cilia seem to be moving a little bit autonomous. Yeah, actually, I want to observe this a little bit longer here. Yeah, yeah, let's go down again with the magnification to get a better overview of what's going on. Yeah. Yeah, I've tried. When you add too much alcohol at once, then basically the alcohol flushes everything and kills off everything instantly. So it's not quite as interesting to observe. Yeah, but yeah, what we can see here is that yeah, also the bacteria, many of the bacteria stopped moving, and the question is of course, maybe we go down further with the yeah. Here is maybe some of the cells have moved either to the edge of the cover glass, or indeed we do not see them anymore because indeed they already died. Here they are. Ah, look at this. Yeah, so, oh, a paramecium. That's the large one over here. 2. Nice. That's a good sign. So this means that paramecia are also starting to divide now. Yeah. So yeah, they were kind of all pushed. Oh, that's a gigantic one over here. They kind of all moved to the center of the cover glass of the slide, because of course the alcohol was approaching from the outside, and here they are, and they're not able to go further because of this gigantic air bubble that kind of blocks the way. Yeah. So the question is this bright field? And the answer is no. This is a so-called the DIC. But if you want to see it in bright field, no problem. This would be bright field now. Okay, so this is the way that you would normally probably see it if you have, yeah. But because of course those microorganisms in many cases, they don't have a color. And yeah, I've got DIC to give it a little bit of color as well. And it looks a little bit also three-dimensional. Yeah. You know, so you know what I'm going to do now. Yeah, I'm going to add another thing a little bit here. And let's see what happens. Oh, I pressed on it, unfortunately. Yeah. But then I simply would like to move on a little bit to to different disinfectant. But alcohol, of course, is very effective. Yeah, and the reason is because it does dissolve the cell membranes. Could I put a dye in your disinfectants to make it more obvious? Actually, it's an interesting idea as well. Have a look, just a second. What I have here is, something that you already know. It's methylene blue solution, which does contain alcohol as well. Okay, so what I could do is I could actually use the methylene blue over here and use this as a as a disinfectant, so to say. and then we're able to see it a little bit better. But what time is it now? I do have to check a little bit the time, because 30 minutes already, and we're just with the alcohol. So I would like to try out the others as well. So this one is now methylene blue. And I'm just going to show you. Here, that's the methylene blue, which does contain all the alcohol. And it should also stain, of course, the cells. Yeah, and we're going to see as it spreads by diffusion across. Yeah, and then maybe it's going to reach the other side as well. And in order to speed this up, this process, I'm going to add a little bit more on the side here. Yeah, but there is already a lot of liquid beneath the cover glass. So for this reason, yeah, it's difficult to, yeah, it doesn't take up much more. So I will use some tissue paper to remove. Look, here it is, the methylene blue. See how it actually stains the cells. And over here somewhere in the center, this is where all of them escaped. Again, I'm kind of happy now that also the paramecia, the larger paramecia, started to reproduce. Yeah. And yeah, I'm going to now do the following. I'm going to now take some tissue paper, and I'm going to remove a little bit of liquid from the edge of the cover glass. You can see how, yeah, it removes quite a bit of liquid. And you see on the bottom, you see already the methylene blue solution. It's a little bit like live staining what we're doing. So here's the methylene blue solution. And maybe some of the cells are slowly going to take it up and maybe also accumulate and build up some of the stain. Yeah. Let's add a little bit more. Ah yeah, you see, those cells here, those dark cells, they have really taken up quite a bit of the stain. Let's take a little bit more. It's actually also a good, how do you say, experiment for live staining, because normally when you add stain, yeah, because in this case it does contain alcohol and it does react with different cell components, it therefore of course kills many microorganisms. But if you get the concentration sufficiently low, you might actually be able to stain them without actually killing them. Yeah. But as soon as they actually get too much of this, then of course it's not good for them. Yeah. So yeah, hydrogen peroxide. Some of you are already waiting for some other disinfectants. Okay, yeah. So let's do another one. I'm going to remove again a little bit some of the liquid over here. So you see how this kind of draws away and pulls away many of those cells as well, along with it. So. Here it is, down here is the blue stain, and they're all trying to escape, and usually they're able to sense, of course, as I mentioned, the unfavorable conditions, and they're going to move away from it. Yeah? So what I'm going to do now is I'm going to add yet more, and then I'm going to, for the other disinfectants, I'm going to try to be a little bit faster, because I do want to try them as well. See how it's starting to stream. I'm going to try now something a little bit different. I'm going to now take a little piece of, and I'm going to press against the cover glass to mix the methylene blue a little bit with the cells. Yeah, they're moving, they're all clustering together in the center, trying to move away. Yep, they don't like it. They don't like it. That's why they're all clustering together, moving away from the alcohol of the stain. Let's go up a little bit here with the magnification. This actually looks also quite interesting. Okay. Yep, we can all slowly see the methylene blue approaching here. So and slowly the cells should be taking up also the stain, and then let's see how they respond. They're really clustered together now. Let's see. See, the paramecium tries to move away a little bit, and let's see if it turns back again. Yeah, there's a comment. It's a good way to increase population density. That's correct. I've usually done this also with salt. So what I've done in the past is I added a little bit of salt solution on one edge of the cover glass, and then they moved away from the salt. And then they start to accumulate on the other edge, and this way I got a very high concentration for filming. Yeah. Yeah. So. Yeah. So let's give it. Yeah, there's actually. Let's try it again. Pushing down a little bit here. Yep. Let's be a little bit patient here. And another recommendation that I have generally is to not use too much liquid in the initial sample, because if you use very little liquid, then any liquid that you add, disinfectant that you add, will be drawn beneath the cover glass much faster, and it's going to mix faster. Yeah? But if there's already too much liquid present, then it's not going to happen. So let's try it again, a little more. So let's be very generous now. Okay. So where is it? Yeah, it's slowly spreading. So here we go. So now I kind of lost them. Oop, this was a little too much, I guess. Where are they now? Let's go down with the magnification. Ah, here they are. Here they are. Yeah, you see that the background, let's do it a little bit away. So that's bright field. Now you can also see those cells here on the bottom that actually started to become stained. Yeah, now we can actually see the effect of the stain much better. Yeah. So I'm quickly going to read, as we watch this, I'm quickly going to read again a couple of comments here. Okay, can you flip the DIC to get another color than the blue background? Yeah, oh, I see. Yeah, yes, that's what I just did. So this is now basically, again, bright field. Okay, and now we're actually able to see the natural colors here. Look, isn't it kind of interesting also if we look into the cells to see all of those little cell organelles inside those cells? Yeah. So I missed the beginning. This is a culture of ciliates. So what I've basically done is the following. I've grown a couple of ciliates and also bacteria in a water sample, and I put them under my microscope, and I'm now adding different disinfectants. I'm still with the first disinfectant, even though I'm already 40 minutes into the live stream. I actually also wanted to try out vinegar and hydrogen peroxide, which I'm going to do, but I'll be doing this a little bit faster maybe than what I've been doing up to this point. I've been waiting now for those cells to be actually stained using methylene blue, which is actually which in this case contains alcohol, which actually also should act like a disinfectant. Okay. And look, those cells up here. Yeah, I'm kind of wondering if they also slowly become stained. Look, this paramecium over here. This one actually, yep, now got its share of methylene blue. Let me quickly check. I'm afraid of using my higher magnification objective because I'm kind of worried that I might get the whole thing covered with liquid. Let me quickly remove. Up, this was a little too much. Oh, here we go again. This is actually a nice example of also staining the cell's contents. So, let me try again, removing any excess. Here we go. So, that is now a paramecium. Yeah. And we can actually see all of the cell organelles, the nucleus in there as well. Yeah. So you can see another interesting effect, and that is do you actually notice that the cell itself, the paramecium itself, is actually much more blue than the surrounding? And that is because the cells tend to accumulate the stain. So they will actually, the stain will react with the cell's contents, and the cell will become more blue than the surrounding. And that's actually the way that you're able to increase the contrast. Yeah. So, I'm just looking quickly on the comments section here a little bit. Yeah. And there's a comment, bacteria movement seem to have stopped altogether. Yes. Look at all of those spiral-shaped bacteria that were really rapidly moving before. Yeah. They basically stopped moving completely. Yeah. And you see that over here there is already some damage to the cell membrane, and the paramecium becomes more and more blue because it starts to accumulate more and more of the stain, which tends to diffuse in here. So let's go over here. See, now we see it again. Yes, here we go, and this was basically it. Some of them are still moving around. Yeah. Something I wanted to show you here, which did not take place, is that actually the cells pop open and spill the contents. On the thumbnail that I showed you, at the beginning of the video, I actually showed you a cell which popped open. So you see that different cells evidently respond differently. Yeah. So look how much they have accumulated the stain. So what I'm going to do is, if you put clean water soon enough after they stop moving, can you save them? I've tried stuff like this already before, especially when they've with salt, and that might be possible, but it's difficult to do because you'd have to exchange everything, the liquid. Yeah. So what I'm going to do is the following. I'm going to go back to I'm going to remove this here. Let's try a couple of other substances here. Especially, I would be really interested, because I did not try this under the microscope yet, some garlic, because garlic is known to kill off bacteria. So let's do one over here. Let's do a second one over here. Let's put a cover glass on each one of them. Number 1 and number 2. So let's have a look again. So let's see, where is the scope view? Here we are again. Okay, so that's a fresh sample. We again have basically it looks pretty much like before. Some more starch grains over here from the food source. And as we can see, the cells are scattered all around, all over the place, and maybe they're going to, again, cluster together somewhere. Yeah. So, okay, so let's give it a try. Again, the desk view, I would like to try now some garlic. So I pressed some garlic and added a little bit of water to make it easier to take up the liquid. Let's see if, yeah. Let's take up some of the garlic juice here, and let's give it a try here. This is indeed something I did not try yet. So let's have a look if this actually works over here. Ooh, look at this change in refractive index. You can actually... And how do they like the garlic? They start spinning around. They don't like it. And let's go up a little with the magnification. Let's have a better look here. And this, I think this was pretty straightforward. Yeah. Yeah, garlic is actually known to kill off microorganisms. There's a substance in there that they really... Oops, look at this guy. This guy's also a little bit out of control. I've seen this quite frequently, that sometimes microorganisms, if they're they start spinning before they die. Yeah, this also happens sometimes when you add salt water. Yeah, and that's it. Yeah, so they kind of go a little bit out of control. So, and over here, yeah, they also start spinning. Yep, garlic is pretty effective. This is actually known that's actually also one of the reasons why, if you want to preserve food, then sometimes you add quite a bit of garlic. Let's remove the arrow again. Yeah. So that was a quick demonstration on the effect of garlic. Let's have a look how the situation looks like generally. Yeah, over here. Yeah, that's as expected. As a matter of fact, what I've done already before, when you're growing microorganisms, bacteria, for example, on agar plates, in petri dishes, then you can actually see that if you place a piece of garlic on the petri dish, that around the garlic bacteria do not grow anymore. Yeah? So because the garlic juice diffuses into the growth medium and blocks the growth of bacteria. Yeah, so you see, this was actually, I don't know, almost more effective than the alcohol. But maybe this is because it simply spread quicker beneath the cover glass. Yeah? No wonder Dracula doesn't like it. Yes. Yeah. So let's move on. Okay, let's try a different disinfectant. Let's move over to the other sample over here. Here again, we should be able to see a few live cells. Here they are. Okay, so that's the other one. Yeah, there are lots of them here. So yeah, so they're still moving around happily. Some air bubbles, and now I would like to try. How do you call this? Let's try hydrogen peroxide, 3%, because this is actually a disinfectant that is used for sanitizing and for disinfecting hands. It's a 3% hydrogen peroxide solution. And I'll quickly, I just show you again what I'm doing here on the desk. I quickly pour out a little bit of this. Okay, so that's some hydrogen peroxide, not the concentrated 30% solution that I used in the last couple of weeks for preparing the insects. Maybe you joined last week. So let's take this here and let's give it another try. Scope view. So let's go there where we're able to this is actually the place where the hydrogen peroxide should be the strongest. They also start spinning. I'm a little bit disappointed because normally what you see is you see oxygen bubbles forming. Yeah, so usually it's quite a little bit more spectacular than this. Yeah, okay. Here there are a few more. I think I'll add a little bit more. Or maybe some of them started to move away again. Yeah, of course. They're over there. On the other side. Here there are lots. Let me try there. I'm gonna add there also some of this. It almost appears that they seem to be moving a little bit faster. Yeah, as they're moving around, of course, they're also mixing the disinfectant with the water. They're of course hiding over there where there are other material as well. And over here, what do we have here? There's nothing. They of course moved away. So sometimes it's a good thing to look at those that are at the border of the disinfectant a little bit. Some of them accidentally happen to swim into it. Yeah, but normally I would have expected some oxygen bubbles, because the hydrogen peroxide, when it breaks down, will actually form oxygen gas. And this is something that we do not see here. Yeah, but still they kind of gather there where they're a little bit more protected. Perfect for Halloween, the slide of horrors. Yeah, I'll add a little more. Yeah, you see how the liquid is streaming? Yeah, and those cells that occasionally tend to touch the hydrogen peroxide, well, that's it. And see, again here, they start to cluster around there where they are a little bit more protected. But those that kind of managed, or those that kind of venture out a little too far, again, look, dividing cell. They seem to go a little bit more uncontrolled. KMNO4, potassium permanganate solution would be great. Potassium permanganate, KMNO4. That's I think has this very pinkish color. It's very oxidizing. So essentially, yes, it's an oxidant, purple color. That's correct. I've once, here are the oxygen bubbles. See, finally. Yeah, basically here what we see is we see the hydrogen peroxide breaking down. The question is why does it do that? Why does the hydrogen peroxide break down here? And the reason is because many microorganisms, in order to protect themselves, have an enzyme called catalase. And this catalase enzyme breaks down the hydrogen peroxide to oxygen and water. So this is kind of a form of protecting itself. Spicy chili, I've not tried today. I do have, but I did try it a few weeks ago. So I'm going to save it up again for a future live stream. I've got this, how do you call this? This how did they call this? One chip challenge. This extremely spicy, chip containing a very spicy pepper. Yeah. So. Yeah, there is a question about how I integrate OBS Studio with the microscope. I will talk about that later on. So when I'm receiving in a couple of minutes the questions, then I'll be essentially also responding to some of the off-topic questions. Yeah. So, yeah, I love your content. One of the best on YouTube. Thank you very much. I'm very happy. I will talk about that as well. Why I'm making content like this. Yeah. So yeah, you see that essentially, the hydrogen peroxide also has done its job here. Yeah, and the microbes, stop moving. Now, every now and then, I do receive comments or emails. Not so much emails, but more or less comments where people are a little bit concerned about the ethics of all of this. Okay, is it okay to kill microorganisms, so to say? As a matter of fact, It was a few years ago where some people actually said that they're going to file a complaint to the animal rights agencies because of microorganisms dying under the microscope. And I gotta be honest with you, no, I do not feel sorry for these microorganisms because they do not have a nervous system. Yeah? It's not, they're single-celled organisms. They do not have a form of self-awareness. They do not have a form of intelligence because they have no nervous system, right? They're not able to feel pain or anything like that. Yeah? And every time when I eat food, yogurt, for example, then basically my stomach acid is killing off billions of bacteria and microorganisms as well. And I just basically here now that, thank you very much, I just received your questions again, okay, which I'll be answering in a couple of minutes. Yeah. So, yeah, that's correct. If you're concerned with the death of microorganisms, you should not wash with soap either. That is absolutely right, right? So what I want to say is basically that if you want to be ethical about protecting the environment and nature, then that is something very important. But direct your ethical energy to the right things. You know, we've got a lot of environmental problems in the world that really need to be addressed. That is the place where we have to start and not worry about microorganisms. Under the microscope. Yeah? So I just wanted to mention this. Having said that, of course, we know that microorganisms, some of them are also quite rare, obviously, and one can, of course, argue to what extent certain microorganisms are also not worthy of protection. One can have a debate like this, but otherwise, from an ethical standpoint, whether they're able to feel pain or not, I have to tell you, this is not something that I'm worried about. Okay, so let's do the following. Yeah, I just wanted to say that because I did receive some complaints some time ago. Yeah? So what else do I have here? With the alcohol, let's try some, before I go to the question, I do want to try a little bit of acid as well. So that is basically vinegar. It's more concentrated than the regular vinegar. I would like to see this as well, how this actually works. So let me, usually I use this vinegar to clean, microscope slides and so on, because to remove water stains and carbonate stains. Ooh, it already smells quite strong. So let's give it another try. Oh, you cannot see that what I'm doing here. Here, this is what I'm doing. So let's give this here another cover glass. And let's have a look again under the microscope. This scope view. Yes, it's another. Keep the rivers, lakes, and soil clean. That will save more microorganisms than not watching them under. That's absolutely correct. Yeah. If you're concerned about the environment, which is important, of course, then let's be concerned about the right things. Yeah. So again, over here we have a lot of cells. They all look the same, obviously. Again, lots of starch here. I'm going to wait again a couple of days. I hope that the paramecia, the larger paramecia, are going to also start exploding in the next couple of days. Look again here. Yeah, for whatever reason they're accumulating here. And let's have another. So I totally forgot which one was now. No, that's not the. I need to smell this. That's the acid. So that's the vinegar. So, and let's have a look here again at the top. And I'm going to bloop. And another one. Okay. Let's see what happens. So this is the place where I started to apply it. And so this seems to be, Yeah, there are a few over here because of course they were flushed away. So maybe those over here will, yeah, experience the acid first. They are hidden here again behind this clump. So, and let's see how effective the acid is. I think I might have to add a little more. See how it kind of flushes everything to the side. And let's wait again a little bit. You know what? I'm going to speed it up again by pressing a little bit on the, mixes everything. Yep. But as soon, also as soon as they get in contact with it. Yep. Oh, okay. Here we go. Very quick. Here we go. This, yeah, acid is also very effective, yeah? As you can probably see. Yeah. So, chlorine dioxide would be a nice thing to watch. I actually, some time ago, what I tried is I got my my hands on a couple of these tablets, chlorine tablets that you put into swimming pools for disinfection. So this actually also, yeah, is quite effective. Yeah. Yeah, but honestly, not very super exciting, I have to say, because yeah, this worked quite fast. So what I would like to do next, I'm just checking a little bit the time here. I'd like to try as a last one. I'd like to try salt water, because salt water is sometimes interesting as well to observe. Because sometimes, depending on the concentration, the microorganisms are able to try to compensate the salt water a little bit, because they can osmoregulate. So here we go. So here we go again. Let's see scope view. Here we go. I need to refocus again. So let's try to find it. Oh, a diatom. Okay, look at this. Nice look. Let's zoom in here. Ah, it's a nice diatom. Okay. Yeah, from the water sample. So let's try to find a place here again. There are a few more. It's right in the center. So okay, yeah, let's try some water. Not water, salt water. Yeah, I just showed to you. I've got this is saturated salt water. Look at the being sucked in beneath the cover glass again. Basically means that the cells are being flushed away again. Occasionally there are some cells here. Let's see where they are. Because what with the larger paramecia, you are able to sometimes see how they actually lose water. They become thinner and thinner. Okay. Let's give it another. Here more. So let's give it another one here. So over here, the salt water starts to move in. What are those very long and thin structures like hair? This was kind of the question. And yeah, these must be filamentous bacteria. So when I see another one, I'm going to show them to you. So this is, yeah, where are they now? They're probably gone somewhere. Oh yeah. Here's one moving around. Oh yeah, here more. Here they are. Okay. Yeah. How much are diatoms impacted by salt water? Well, it depends on the diatoms, of course, but generally if you've got freshwater diatoms, and when you add salt water, of course, they're not gonna like it, and vice versa, by the way. So microorganisms are generally quite sensitive to salt changes in salt concentration. They have to adapt to different salts. Sometimes they are able to a certain degree adapt to it. But if the concentration is too high, like for example over here, this is saturated salt water, then this is not something that they experience naturally in their environment, and therefore they're not able to handle it. So this is the thing that we have to keep in mind. If the salt concentration or pH, the acidity, is too extreme, what they've never encountered in their natural environment, then they're generally not able to handle it, because there is no need to be able to handle it, because it's not part of their environment. Yeah? And that's actually also one of the principles of why certain substances are able to kill certain microorganisms, because these substances are not present in their natural environment. Yeah? The very long filaments, yeah, there can be let's see. I don't know if you're talking about, let's say, over here. I don't know if you're able to see those. If you see those very tiny, thin filaments over here, yeah, many of them are actually filamentous bacteria. They're way too thin for hair or for microplastics and so on. These are most likely filamentous bacteria. Yeah. So, and as a matter of fact, what we can do is let's just, for the fun of it, let's go into phase contrast. And here we go. Yeah. And that's basically 40 times phase contrast, and that's very typical for bacterium. I know bacteria, you might be thinking that, yeah, why should they be so long? But indeed, there are filamentous bacteria. Sometimes maybe there are multiple of them sticking together, right? But those tiny things that you see floating around, these are definitely also bacteria. But there can be bacteria that are actually that long as well. Or maybe there are several of them joined together. Yeah. Yeah, so that's not phase contrast. Yeah. Yeah. So. Yes, it's a little bit too much, the magnification, so let's go back again. So, but the salt water seems not to have reached them yet. And yeah, because what they do is again, they kind of tend to move away. Yeah, and they're whizzing about here. What time is it? Oh, it's already 1 hour and 9 minutes. Yeah, again over here you see those long, fine structures. Yeah, I'm quite certain. Yeah, these are bacteria. So, yep, and the largest ciliates seem to be, I don't know, hiding somewhere. Again, a lot of starch grains that you see here in the background everywhere from the food that I added. Yeah, here they are. A few of them at least. Yeah, yeah. So let's mix it again a little bit more. And yep, you know what? I think I'm just going to leave it here like that. We're going to wait a little bit until the salt does its action. I think there are other microorganisms that are a little bit better suited to observe with salt water, because then you can actually observe plasmolysis quite well, how they are actually losing water and become thinner. They change their shape, but we can already also see that over here they seem to be struggling a little bit. Yeah, they're not quite as motile anymore as they used to be. Yeah, and yeah, they also become a little bit thinner because they're losing more and more water. Let's have a close look at those two here. Yeah, you see that many of the bacteria in the background also, well, some of them are still moving a little bit. Yeah, but some of them, yeah, not anymore. So I think you get the idea. What I'm going to do now is I'm going to have a look at some of your questions again. Yeah, maybe I will. Yeah, I received those questions a few minutes ago, and I will answer those that are not on the sheet here a little bit later. Let's do it like that. So I need to clean up my desk here, and so let's have a look. And then I also would like to have another later on after this part. I'm going to ask you another question because I do need a little bit feedback from the community. What's the alcohol percentage you are using to disinfect? As a matter of fact, I think it was 96 or 97%. Why exactly that? There is a certain percentage of alcohol because alcohol will attract moisture from humidity from the air. And I think 96 or 97% is the concentration where it's in balance with the moisture in the air. If you can get yourself alcohol of a higher percentage, 99% even. But the problem with that is that as soon as you open it up, it's going to absorb moisture from the air, and then it's going to go again to 97%. Yeah. So for this reason, because we're mixing it anyway with the water on the microscope slide, the percentage changes. But as I said before, it's really important to know that for disinfecting in the microbiology lab, we always use 70% alcohol. Yeah. Off-topic question: is there a problem with the form? The thing is the following: generally, I'm receiving, as I mentioned already, a lot of spam registrations, a lot of them. And I therefore included a question, a microscopy question of why do you want to join the forum? The problem these days is that even AI is able to answer those. So I'm getting a lot of spam registrations or request that you send me an email if I did not unlock you. So you email with your login, name that you registered, and then I can unlock you. Yeah. So I generally will do this fairly quickly. Yeah. For whatever reason, many of my lab mates hate pipetting, but I love it. Okay. Yeah. This is. Why not just talk a little bit about that? For those of you who don't know this pipette, I think it's very convenient. This goes from 5 to 50 microliters. You can adjust it by turning this here, right? And then if you want to take up a liquid, you basically push it in. You go into the liquid and you release. Yeah. And for pushing the liquid out, you push it all the way in. Okay. This kind of blows out even the last bit, and for throwing off the tip, you push the other button over here, and then will be kind of pushed off. It's a disposable tip, and if you need new tips, of course, then you can always do something like this. Yeah, up, and then you have a new tip, and you can throw it off again. Yeah, so this These were actually fairly cheap. The better ones there cost several €100 or so, but this was actually kind of cheap. Yeah. I've recorded several ciliate deaths in my live stream by accident. Yes, I already commented on that. Yeah, I've recorded paramecium conjugation 2, makes you wonder which one is controlling the movement direction as they cruise around. Yeah, if one is stronger. Yeah, in conjugation, two cells will join together and they will exchange DNA and then they're swimming around. By the way, even when they're dividing, both of them are swimming around together. Yeah, so somehow it's some kind of a coordinated movement here. Just saying hi again and thank you for sharing your time and interests with us. Although our planet hasn't had these many people on it, its structure isolates individuals. Yep, but the internet brings us all together again. And here are some identifications. Thank you very much. Okay. Yeah. Paramecium with acetic acid. Is it possible to see the trichocysts released? I don't know. Trichocysts are essentially structures that are released to defend themselves. So sometimes you can actually see them that they kind of all of a sudden dump them out, and these are like tiny little needles which basically are used for this defending themselves. Yeah? But if it dissolves the membrane, might it not do the same with the nucleus membrane? Generally, I think so, yes. Generally, what will happen is that the outer membrane, the membrane of the cell will be dissolved first. And then when the alcohol reaches the other membranes, then they should also be dissolved because the membrane, the cell membrane is actually made of a lipid, yeah, phospholipids. And those lipids are somewhat hydrophobic and therefore alcohol is able to dissolve them. Yeah? So this is the reason why actually when you say the nuclear membrane, yeah, I think it should also be dissolved if it is actually comes into contact with alcohol. Yeah. If you put clean water soon enough after they stop moving, can you save them? I've also tried this before. It is possible if you're able to quickly remove whatever substance you've got there. And it depends also a little bit on the concentration of the substance. So I've tried this, I think, with a little bit of salt water once. So if you quickly remove the salt water and add regular water again, then you might actually be able to save them again. But it depends, how much the damage has already been done to the cell. Yeah? Will you try honey? Oh, that's interesting. Versus saturated table sugar solution as a germicide. It's the following. If you have that's going to be, for practical purposes, it's going to be difficult because it wouldn't mix very well with liquid. However, it is correct that honey containing a lot of sugar and table sugar is actually also able to preserve. So for example, jam, for example, and honey can be stored even unrefrigerated because the high concentration of sugar will actually cause the cells, bacteria which fall into the jam, for example, or into the honey, to lose water very quickly. And this basically means that they're not able to retain water, so they're going to dry out essentially because they're going to lose a lot of water by osmosis. Yeah? So, but adding honey or a saturated sugar solution which is very thick and viscous, I think I can try that. But I think it won't work by simply adding the solution next to the cover glass because it simply will not be sucked in because it's too thick. Yeah? Maybe colorful fruit or vegetable juice as a dye, like fresh beet. This is not a new idea, but I think that the concentration has to be sufficiently high. And I think that's the other important thing, that whatever stain you're using, it should react with the structures that you want to stain. So I can imagine that there might be certain vegetable juices. They usually contain anthocyanins, which are in red beet. They are pH sensitive. I don't know to what extent they're taken up by microorganisms, but that would be indeed an interesting thing to try out. Yeah? By the way, just a second. Look what I've done here. It's a little bit off topic, but those strips that you see here are actually red beet. That's a filter paper with red beet, with the intention of actually making pH strips, simply for experimental purposes. Yeah. So the idea is can you not maybe use also the red beet dye to stain microorganisms? I never tried that, but just realized the following. I've not tried this either. Where's the? Just a second. Yeah, that's the acid. You know what I'm gonna try? I'm gonna try if this actually works. I'm going to try to put some of the acid on this strip here and see if this actually is able to change the color around. Yep, it does work. Okay, see, when you add it, it actually turns red. Yeah? So this is kind of like a homemade pH indicator strip. Yeah? And I don't know if you're able to see it quite well, but I'm actually able to see it quite well. It turns from purple quite to red. Yeah. So this actually does work. Huh? So let's have a look here. Let's put this away again. Potassium permanganate. Yeah, I talked about that. And why would it produce hydrogen peroxide? It doesn't do that in the bottle either. I do not know now the how do you say, the context of this. What would produce hydrogen peroxide? I think you meant to say oxygen gas. And maybe this question refers to the question of why there are oxygen gas bubbles present when you add hydrogen peroxide to some of the cells. And the reason is because some cells actually have the enzyme catalase present to protect itself from highly reactive oxygen radicals, as it's called. And the hydrogen peroxide is also broken down by this catalase. Yeah. So as a matter of fact, it's the presence of the catalase that will actually break down the hydrogen peroxide very quickly. The hydrogen peroxide in a bottle will slowly also degrade, but what I heard is that for this reason they have some stabilizing substances included in the hydrogen peroxide bottle so that this natural breakdown does not happen quite as quickly. Yeah. So I'm going to now do the following. I'm going to go over to the chat and I'm going to, because there are many more comments here. Okay. At Microbehunter, we love your constant curiosity. Okay. Yes. How do you donate? Oh, thank you very much. If you want to donate, there should be the possibility to, I do have a Patreon account, and there should also be the possibility to donate over the live stream. If you donate money, then I will use this money to buy microscope slides, which I'm going to share with you again in the live stream. Okay, I've done this also in the past already. Yeah. Magnetotactic bacteria. I've tried to find them as well. Was also not successful, unfortunately. So this is the forum. A conclusion of this live stream: shower with garlic. Yes. Yeah. So because garlic does kill bacteria apparently quite effectively. Yeah. So at least it makes food store a little bit longer. Yeah. I recently got some water from the ocean, read up, and you can get saltwater culture media for pretty cheap. Okay, hopefully I can get a good culture of diatoms going. Ah, yes. Some diatoms, what I also read somewhere, you have to be, it's not only that you have the proper surrounding medium, that's important, but some diatoms, they need a surface to grow on. Right. So what I heard sometime is that you might want to put maybe some tissue paper or some cotton towels into your diatom culture, because some of them wanna grow on the surface, and cotton towels and tissue paper, they have a large surface area for diatoms to actually grow on. Yeah. Is it a colony or a single bacteria? I don't know if what you're referring to right now, but it could be that that you refer to those filamentous bacteria. Sometimes it could be that they might appear as one filament, but actually that there are many of them together. Yeah? We don't refer to them in this case either as a colony. We only refer to them as a colony when they're growing on a Petri dish, yeah, and where you can actually see them clumped together. Yeah? I've read somewhere that blood has the same salt concentration as seawater because we evolved from the sea. Is that true? I'm quite sure not, because the seawater is much saltier. Okay, and physiological salt solution, that's basically the one where is a 0.65 percent. 0.65% is salt in water is approximately the salt concentration of our body. And seawater or marine water can have, I don't know, let's say up 3%, 4%, and even more. Yeah. So seawater is much saltier, and it's like this that yeah, even if we evolved out of the sea, this does not mean that essentially our body is not able to adjust itself to different salt concentrations called homeostasis. So where essentially our body is trying to maintain body internal constant conditions, and regulating water balance is extremely important. Yeah, so this is something that the body does. That's why our kidneys especially are responsible for regulating the salt water the salt amount in our blood. If you're interested in what happens when you mix salt water with blood, some months ago I did make a video on that, and you can actually see that the red blood cells, they will actually lose their shape and they shrivel up and shrink when you add salt water. Yeah, so I did make a video on this. Yeah. What salt concentration are you using? I expected a bigger osmotic shock. It's a concentrated salt water because there is salt on the bottom here. However, the reason why the effect was not quite as strong is because now when you are adding, let me just take a slide here to illustrate this. Right, when you have a slide here with a cover glass in the center, let me just also take a cover glass to show you where do I have my cover glasses. Okay. If you just take a cover glass here, so you cannot see it anyway. Then basically the thing is the following: when I add salt water on the side or any other liquid, then it will only move beneath the cover glass sufficiently quickly if there is not enough, if there's not too much water present already. Right? If the cover glass is kind of swimming on the surface, then the salt water or whatever liquid I have will also stay on the side. So the effect is therefore not quite as strong because the cells basically did not get into contact with the salt water. Yeah? So that's why I pushed the cover glass with this little stick here to mix the liquid on the side better with the liquid which is already present beneath the cover glass. Yeah? So this is, I think, the reason. What happens, however, also is if you do apply a little bit of the salt water on the side, and if you wait long enough, half an hour or so, then the salt solution will diffuse across, and then you're also going to see that the microorganisms will start to build up on the other side. Yeah, we didn't have time to do that today because it does take quite a bit of time for the salt solution to diffuse. Yeah. We could gradually change a freshwater diatom to saltwater one by slowly changing the salt content during a couple of years. What you'd have to do is, in theory, so again, I'm going to read this a little bit slower. Could we gradually change a freshwater diatom to a saltwater one by slowly changing the salt content during a couple of years? And the answer is if you want to do that, essentially what you're doing is you're doing evolution, so to say. You cannot do that with individual diatoms, but if you have a whole population of diatoms which is continually reproducing, and when you're changing the external conditions, what you're doing is you're applying a so-called selective pressure on the diatoms or on whatever microorganisms you're investigating. And indeed, this can mean that you are directing let's say the salt tolerance into a certain direction. Yeah. As a matter of fact, I've been doing something similar at the university with bacteria. If essentially what we did, this is a little bit was a little bit dangerous. We added mutagenic substances, basically substances that cause mutations. We added those to bacteria and tried to make them a little bit more resistant to certain substances. Yeah. So it is possible to do that. I don't think that this is scientifically very interesting because things have already been done, are always done. As soon as you, for example, when you breed certain animals or plants, what you're essentially doing, you're also applying a certain, you're choosing which ones are able to survive. Yeah. As a matter of fact, this is actually an interesting point as well, because I read somewhere that in dishwashers, which basically are quite frequently very hot, obviously, you can find a lot of heat-tolerant bacteria, and they actually, over the many years that you're using a dishwasher, that's basically they've become, as a population, they've become more heat-tolerant. Yeah. So if it's possible with bacteria becoming heat-tolerant, why not also with diatoms becoming more and more salt-tolerant? Yeah. Yeah, I think this is were most of the questions already. Yeah, I think I answered all of those. Why doesn't bleach kill all germs? That's an interesting one. I will quickly tell you the following. It depends, of course, on the bleach concentration, but that's an interesting one. If you have got a disinfectant which kills, let's say, 99.9999% of bacteria, and if you've got billions of bacteria present, if you just do a little bit of math, then there will be still many, many bacteria that survive. Yeah. So especially when you're dealing with very, very large quantities of bacteria, even a 99.99% effectiveness of removing the bacteria is still going to allow many bacteria to survive. Yeah. So this is a little bit, you have to make sure that if you want to really kill all of the bacteria in a certain place, you really have to make sure that the disinfectant is able to reach all places equally. Yeah. And so that's, and we've seen this a little bit here also in the demonstration, that they've added some kind of disinfectant on the side of the cover glass. And some of the microorganisms were hiding somewhere in the center between some clumps of algae or food or whatever was left on the slide, because this is the place where the disinfectant which was not able to reach. Yeah? And this is where the microorganisms started to accumulate. Yeah, so this is a little bit the thing here, that when disinfecting substances, you have to be really careful that this is done properly. Yeah. So I think this was actually, yeah. Yeah, that's an interesting one here. It relates a little bit to the ethics. When I worked at the zoo, a lady came up to me at the bobcat cage and said it was wrong to cage them. She was wearing a fur coat. I know it's maybe a little bit off topic, and I know that it's important to keep animals well in a zoo and so on, but if you do not give people, or especially children, a possibility to see zoo animals in real life, yeah, and experience that, then how are they supposed to learn how to love and respect nature and animals? That's a little bit the thing that I think it's important that children especially are exposed to these also to animals. Yeah, and I think the zoos these days are a far cry to what we had back in the, let's say, 19th century, when animals were really kept in very small cages. But generally, if you've these days, I think zoos are spending quite a bit of effort in actually making it animal friendly. At the same time, of course, it's not freedom that they have, right? Yeah. So yeah, I think what are the 0.0001% survivors, cysts or protected organisms? Oh, I get it. Okay. That's an interesting point. I just mentioned that even if you basically have a large quantity of bacteria and you're basically killing off, let's say 99.999%, that you still got a lot of them surviving. Why are they surviving? It could be indeed that some of them might be cysts. However, that doesn't mean necessarily that. It could be because in a natural population, you always have a genetic diversity and genetic variability. And some organisms are always going to be more tolerant than others. You always have this distribution curve, this Gaussian distribution curve. At a certain concentration of disinfectant, some bacteria might die faster or easier than others, simply because there is a natural variation present. Yeah? And only if you go up very high with the concentration, if you really make sure that all of the disinfectant reaches all parts, only then you're probably able to get, let's say, close to 100%. Yeah, but if you've got a very, very high concentrations of bacteria, then, and if you just dump some disinfectant over it, then maybe only the outer ones are of the colony, let's say, are able to be killed off, and maybe the inner ones are able to survive more. So you also need to make sure that there is sufficient time for the disinfectant to act. Or for example, in a microbiology lab, what you do is you don't just dump disinfectant over it, but you autoclave them. It means you have to at least heat it up to, I think, 130 degrees or so for half an hour to make sure that really pretty much everything's killed off, including any endospores, which are basically like cysts, which are able to be quite heat tolerant. Yeah? So there are indeed in the microbiology lab, there are certain protocols that you have to adhere to make sure that the microorganisms are really properly disposed of and killed off. Yeah? So what I'm going to do now is I think I'm going to quit again, but no, I'm not. I'm not. I'm going to ask 3 minutes because I do have a question, and I would like to ask the community for a little bit of help and support. The thing is these live streams are quite more or less watched by around 1,000 people, 1,100 people. But many of my other videos in the channel are really not so successful, and I don't know why. So maybe the topics are not quite as interesting, or I don't know what it is. Maybe some people are saying that I'm too scientific, I'm throwing around with too many biological terms, which I actually didn't think, but maybe that could be the opinion of some. So if you give me some feedback of what you're interested in or how I can improve the videos, I would be very thankful for that. Okay. A couple of months ago, some of you suggested I should not include music into my videos because this was considered distracting. Yeah, please, feedback is always very welcome. Also concerning the topics that I should cover. Yeah, especially if you are new to this channel here, and if you're not so much into microscopy yet or biology yet, it's very helpful for me to know a little bit more of what would interest you. Okay. Is it possible to make in the future a video about the identity of the cell contents? And the answer would be yes, I could do that. However, and this has been a request that's appeared a few times already about the contents of the cell. However, the thing is the following, that many of the things we're not able to see with a light microscope. We would need an electron microscope, and I don't have that. Yeah, so this is a little bit the problem. I can still talk about them by kind of doing a little bit like a biology lesson and kind of theoretically showing you what you're able to see inside a cell. But sometimes but you know what? I can still show you the things that we are able to see. Why not? Yeah. But some of the structures we're not able to see, unfortunately. Yeah? Again, a comment here. I'm less interested in videos that have topics that were covered in the previous live stream. It feels like I have already seen this, although I really enjoy the streams and video. Yeah, that's another thing that I know. There is unfortunately only that much. I know it's over time, it might become a little bit repetitive. Yeah. And that's indeed also one of the things where I'm struggling a little bit is to come up with new ideas, new variations. But of course, sooner or later, you've more or less seen it all. I still hope that even if some of the things are a little bit repetitive, that I'm always able to add a little bit of new info to it and have a little bit of a new take. Yeah. So that's a little bit something that I would like to also mention. And one of the reasons, and that's also a comment that I would like to, promised myself that I would like to make today, is one of the reasons why I make those live streams, and is also because I want to, how shall I say this? Biology sometimes can be quite theoretical as well, right? And with these live streams, I kind of want to reconnect a little bit with nature and the environment. I am a biology teacher, but many things that I actually teach are quite theoretical, right? And I think that microscopy and nature observation can kind of maybe add a little bit back this original, how do you call this? I don't know, I'm looking for the word, enthusiasm maybe, or feeling of mystery maybe, or it's a little bit when you're watching the stars, right? you kind of feel a little bit impressed of seeing the Milky Way and the stars, kind of a sense of wonder. And this is a little bit something that I would like to recapture as well with these live streams here. Yeah. So I'll be reading your comments. Yeah. And I would like to thank you very much for those. If you have any comments or so or suggestions, then please do feel free to not only post them in the comment section, but also in not only in the live comments, but also in the comment section below. Would be very thankful for that. And yeah, I'm reading a little bit here as well. Love to see a video of yourself taking water samples in your local pond. I actually did prepare something. Okay, I'll make a video of this. I'm going to call it quits for today. Yeah, and yeah, I'm going to maybe include a little bit some more theoretical stuff in the future as well. Maybe from the stuff that I've been doing when I was still working at studying at university. Some more detailed microbiology maybe. Okay, enough for today. I'm already a little bit over time here. Hope you liked it, and see you again. All the best. Bye-bye.